Hello friends, today in this video we are going to discuss about the basic microbiology lecture 3, and our main topic here in this video is bacterial cell wall structure. So our topic index, here we will see our topic index. So first topic is bacterial cell wall structure, and next is antibiotic on bacterial cell wall. That means the antibiotics, how the antibiotics affect the bacterial cell wall. And next, the archaeal cell wall structure. That means what are the difference between the bacterial cell wall structure and archaeal cell wall structure. And the last point is cell wall less bacteria, means how the cell wall less bacteria survive in environment without their cell wall or rigid structure. Okay, so all we will see in this video. So first thing is bacterial cell wall structure. So first we will discuss about the bacterial cell wall structure. So suppose this is a bacterial cell wall, and this outer layer is known as capsule or glycocalyx layer. Okay, and inside this capsule there is a compound. This is known as the structure, that is known as the cell wall. So inside the capsule or glycocalyx layer, the cell wall is present in a cell, and inside the cell wall there is a plasma membrane which is present outside of the cytoplasm. So here the cytoplasm is there, and this cytoplasm, that contain the DNA or ribosomes, which are the cellular compounds. So this all these cytoplasmic items are covered by the plasma membrane, and this plasma membrane is covered by the cell wall. So in this video we are going to talk about the cell wall structure. So now let's move to the cell wall structure. So if we zoom this cell wall structure, what we can see? We can see this type of structure. So this is our plasma membrane or cell membrane, and this is the cell wall structure. And for gram positive bacteria, the cell wall structure, which is made up of peptidoglycan, this cell wall which is made up of peptidoglycan is mainly thick. Okay, so this in gram positive bacteria peptidoglycan layer is thick, but in gram negative bacteria the peptidoglycan layer is comparatively thin to gram positive bacteria. And another difference is in gram negative bacteria a outer membrane is also present in gram negative, but in gram positive bacteria there is no outer membrane present here. Okay, so these are the main difference between gram positive and gram negative cell wall, gram negative bacterial cell wall. Okay, so now let's see the peptidoglycan layer of cell wall. So here we can see this is the peptidoglycan layer which made our bacterial cell wall. Okay, so this peptidoglycan layer here which is present in cell wall, that here also some another compounds are there that are known as the teichoic acid. These are the teichoic acid, and if this teichoic acid only present in the cell wall, then it is known as simply teichoic acid. But if this teichoic acid is started with the plasma membrane or lipid layer and then go to the peptidoglycan layer, then it is called the lipoteichoic acid, because of lipid, because it is attached with the lipid molecule of the plasma membrane. So two types of molecules there, that is teichoic acid and lipoteichoic acid, and the space between the peptidoglycan layer and the plasma membrane layer, this space, the inside space, is known as the periplasmic space. Okay, the space between the peptidoglycan layer or cell wall and cell membrane is known as the periplasmic space. Okay, so this is all about the gram positive bacteria, and now we will see the gram negative bacteria. So here in gram negative bacteria, this is the outer membrane, this is the peptidoglycan layer, and this is the inner membrane or cytoplasmic membrane, and the space between the outer membrane and inner membrane is known as the periplasmic space. So here you can see that the periplasmic space in gram negative bacteria is more, because the because the peptidoglycan layer in gram negative bacteria is very less or very thin. That's why the periplasmic space is more in gram negative bacteria, and also in outer membrane of gram negative bacteria some different types of compounds are there. They have the lipopolysaccharide. This means this outer membrane of the outer, this in outer membrane some lipid molecules are there, and with their lipid molecules some carbohydrate or polysaccharides is attached, and that's why it is called lipopolysaccharide. Lipid plus polysaccharide. Okay, so they are called lipopolysaccharide. And another, some porine proteins are there. These are some porine proteins, or also known as the channel protein. By through some different outer molecules can get into the inside of the cell. Okay, so these are the different compounds of gram positive cell wall and gram negative cell wall. And now we have to talk about something which are present in gram positive bacteria and gram negative bacteria. So here we have to change the color. So in periplasmic space, this compartment provides an oxidizing environment that contains degradative enzymes where many protein structure can be stabilized via disulfide bonds. So in periplasmic space, different types of protein can be stabilized here by disulfide bond. This is the one point for periplasmic span. And another thing which is provided by the periplasmic space, that is the presence of this cell compartment, that is periplasmic space, in bacteria allow them to sequester potentially dangerous degradative enzymes such as RNase and alkaline phosphatase. Like RNase and alkaline phosphatase, which are dangerous for the bacterial cell. So if they go into the cell, then the degradative enzymes which are present in the periplasmic space help to degrade this RNase and alkaline phosphatase and ultimately provide protection to the bacterial cell. So this is the role of periplasmic space. And now we will discuss about the teichoic acid. So what is teichoic acid? We have already discussed that teichoic acid is present in the gram positive cell wall, and it provides stability because this structure is present inside of the peptidoglycan layer. So it provides a compact or cross-linked structure inside the um, inside the peptidoglycan layer, and it provides a rigidity in the peptidoglycan layer. So it provides stability and helps in passage of ions like magnesium. So it helps in passage of ions inside the cell and outside the cell through the cell wall or peptidoglycan layer, and give negative charge to the cell. This teichoic acid is itself a negative charge, so it gives a negative charge to the peptidoglycan layer. Okay, and it only present in gram positive bacteria. Already we have talked about this, that the teichoic acid only present in gram positive bacteria but not in gram negative bacteria. Okay, so move on to the next part. So here we will see this peptidoglycan layer more specifically. Okay, so we have to zoom this peptidoglycan layer, and what we can see. So here this is a cell wall structure, and if we zoom this structure of a cell, we can see this is the plasma membrane. This is the plasma membrane structure, and outside of this plasma membrane the cell wall or peptidoglycan layer is present. Okay, so this cell wall is made up of this peptidoglycan layer. So this whole total layer is a peptidoglycan layer. This one is another peptidoglycan layer. This one is another peptidoglycan layer, and also this one is the another peptidoglycan layer. And each of these peptidoglycan layer is composed of two different molecule. One is NAG, means N-acetyl glucosamine, and another is NAM, that is N-acetyl muramic acid. Okay, so and this one peptidoglycan layer is attached with the another peptidoglycan layer by peptide linkage. This is the tetrapeptide which is attached with the NAM, and this is another tetrapeptide which is attached in the NAM, and these two tetrapeptides are cross-linked with each other and provide a rigid structure or compact structure to the cell wall. Okay, so how this structure provide a rigid or compact structure, that we will see in our next slide. Okay, so this is the peptidoglycan layer. This is the one peptidoglycan layer. This is the second. This is the third peptidoglycan layer. This is the fourth peptidoglycan layer. All are the peptidoglycan layer, and each of these peptidoglycan layer is composed of NAG and NAM, and this NAG and NAM alternatively go whole of this peptidoglycan layer. So what is peptidoglycan layer? This peptidoglycan layer or murein is a polysaccharide consisting of sugar and amino acid. This NAG, NAM, these are the derivative sugar derivatives, and amino acid means these are the peptide bonds or peptide polypeptides which are made up of amino acids. So peptidoglycan layer is a polysaccharide consisting of sugar and amino acid which is present outside of the plasma membrane and give a rigidity to the cell. Okay, so now we will see what is NAG and what is NAM. So NAG means N-acetyl glucosamine and NAM means N-acetyl muramic acid, and now we will see the structure of NAG and NAM. So how their name defines their structure, that we will see. So NAG is N-acetyl glucosamine. So here the gluco, this is the glucose. So gluco means glucose. So this is the glucose structure and it is amine. So glucose and amine. So here this is the amino group present. So this is the glucose and this is the amino group, and altogether this is known as glucose amine. Okay, and next is acetyl. This is the acetyl group. Okay, and this acetyl group is attached to the amino group with the N or nitrogen. That's why it is known as N-acetyl. So altogether it is known as N-acetyl glucosamine structure. Okay, so its structure is defined by its name. Okay, so next we will see the N-acetyl muramic acid. So muramic acid is made up of lactic acid and glucosamine. So glucosamine means this is the glucose and this is the amine. Okay, so this is the glucose and this is the glucose and this is the amine, and how the muramic acid is formed. So muramic acid is glucosamine and the lactic acid. This one is the lactic acid. Okay, so this is the muramic acid, and what else is there? That is acetyl, N-acetyl. So here acetyl group is present which is attached to the amino group N. So that's why it is N-acetyl muramic acid. Okay, so muramic acid is made up of lactic acid and glucosamine, and here this is the N-acetyl muramic acid. Okay, so this is the whole structure of muramic acid, and now this one and this one will bind with together and form the peptide layer. How? So this is the first carbon of NAG and this is the fourth carbon of NAM. So they bind with each other, and this is the beta position of group. So this is the this one is the fourth carbon. So here the 1,4 beta linkage is present. 1,4 beta 1,4 linkage is present. Okay, how? Here you can see this is the beta 1,4 linkage. Okay, and here also one thing, that is with the NAM structure the tetrapeptide are attached with the lactic acid group. Okay, so here you can see NAG NAM. Here in this way the peptidoglycan layer is formed, and with the NAM tetrapeptide or 4 amino acids are attached with all this NAM, and one tetrapeptide is attached with the another tetrapeptide by cross linkage and provide a rigidity to the cell wall. So how this cross linkage formed or peptide bridge is formed, Now we will see here. So these are the peptide 1 or amino acid 2, amino acid 3, amino acid 4 in one peptide layer, and this is another peptide layer or peptidoglycan layer, and here this is another NAM, and here the amino acids are 1, 2, 3 and 4. So with the third amino acid of one NAM and with to the fourth amino acid of another NAM is attached with a peptide with a linkage or cross link between peptide linkage between them. So in this way the peptidoglycan layer is stabilized. So what are these amino acids and what are these peptide linkage, so that we will see in next slide. Okay, so what we can see here, so here we will see this is the NAM, this is another NAM of different peptide layer, and with the NAM different amino acids are present. So what are those amino acids? So first amino acid is L-alanine, then D-glutamic acid, then L-lysine, then D-alanine. So we can remember it as L, D, L, D, LD, LD, and the first one is alanine and also last one is alanine. Okay, so in this way we can remember L-alanine, D-glutamic acid, L-lysine and D-alanine. Okay, also in different peptide layer, also here the NAM, in the NAM also present, that is L-alanine, D-glutamic acid, L-lysine, D-alanine. Okay, and with the third amino acid, that is with the lysine of one NAM to the alanine, D-alanine, the different, that is the fourth, that is the fourth amino acid, and this is the third amino acid, they create a bond or peptide linkage with the glycine. So the pentaglycine bridge is formed. Okay, so peptidoglycan NAM of one peptidoglycan layer and NAM of another one peptidoglycan layer is bound to each other by the pentaglycine bridge with their tetrapeptide. Okay, so this is the whole thing. And now another thing we will see here, in gram positive bacteria we can see this is the NAM, this is the tetrapeptide, this is the another layer NAM, this is the another tetrapeptide, and both of these tetrapeptides, this is in the with their third amino acid to the fourth amino acid, cross linkage is done by pentaglycine molecule. Okay, but this thing only present in gram positive. In gram negative bacteria, in gram negative we can see here the DAP, instead of L-lysine there is DAP molecule is present. DAP means diaminopimelic acid. Okay, and this DAP, that is the third amino acid, is bound with the D-alanine of another NAM, that is the fourth amino acid. So they linkage with the DAP and D-alanine instead of L-lysine and D-alanine, L-lysine and D-alanine. But here no pentaglycine is present in gram negative bacteria. Pentaglycine is only present in gram positive bacteria, not in gram negative bacteria. Okay, because this glycine structure provide this penta bridge or pentaglycine bridge provides a very cross-linked structure or very compact or rigid structure to the gram positive bacteria, because the thickness is very because their thickness is very high or they are more thick in gram positive, but in gram negative bacteria the layer of peptidoglycan is very less, so they don't need a very compact or very rigid structure of peptidoglycan layer. So that's why they only have a general linkage, but in gram positive bacteria we get a pentaglycine bridge. Okay, so move on to the next. So now we will see how the peptidoglycan layer is synthesis. So this is the NAM, and sorry, this is the N-acetyl glucosamine or NAG, and this is the N-acetyl muramic acid or NAM. And now we will see this is the whole peptidoglycan layer, one peptidoglycan layer with the tetrapeptide attached with the NAM. Okay, and this is the another peptidoglycan layer where this is the tetrapeptide attached with the NAM. Okay, and now this tetrapeptide and this tetrapeptide will bound to each other or cross-link to each other by the enzyme that is known as transpeptidase. This transpeptidase enzyme will cross-link between two tetrapeptide with the pentaglycine molecule. This pentaglycine molecule provide the cross-linking. Okay, by pentaglycine bridge. So this gives a rigid structure to the peptidoglycan layer or to the cell wall. Okay, so now we will see how this peptidoglycan layer give a rigid structure to the cell wall. So this is a whole peptidoglycan layer. This is the first peptidoglycan layer, and this is the tetrapeptide. These are the tetrapeptide, and this is the next second peptidoglycan layer, and the cross linking is done by the tetrapeptides in there by the glycine pentabridge. Okay, and this one is the third peptide layer, and another glycine pentabridge will form, and this is the fourth peptidoglycan layer, and glycine bridge is formed by the tetrapeptidase. Okay, so in this way the peptidoglycan layer or cell wall form a compact rigid structure by the cross linking through glycine pentabridge. Okay, pentaglycine bridge. Okay, so now we will see the characteristics or difference between cell wall of gram positive and gram negative. So first thing is the peptidoglycan layer. So peptidoglycan layer is thick. So this is a thick peptidoglycan layer, and in gram negative bacteria the peptidoglycan layer is thin. Next one is teichoic acid. Teichoic acid is present in gram positive bacteria but not in gram negative bacteria. Next thing is lipid. Lipid is, we have seen that in gram negative bacteria the outer membrane, here we can see, in the outer membrane the lipid molecules are present in the cell wall of gram negative bacteria. So the lipid molecules are present in as a lipopolysaccharide in the outer membrane of gram negative bacteria but not in gram positive bacteria. So very little in gram positive bacteria. The next one is outer membrane. So outer membrane is present in gram negative bacteria but not in gram positive bacteria. Periplasmic space is very less in gram positive bacteria because the in gram positive bacteria the peptidoglycan layer is very compact and thick, and they only have a cell membrane and the peptidoglycan layer. So the space between this peptidoglycan or cell wall and the plasma membrane is very less. This one is very less. So that's why periplasmic space is mainly absent in gram positive bacteria, but in gram negative bacteria the space between the outer membrane and cell membrane, between these two structure, the peptidoglycan layer is very less, very small. So there, in that portion the space is large or comparatively large in gram negative bacteria. So that's why periplasmic space is mainly present in gram negative bacteria. Next thing is cell shape. So as the peptidoglycan layer have a very thick peptidoglycan layer, or in gram positive bacteria peptidoglycan layer is very thick, that's why it gives a, always gives a rigid structure, but in gram negative bacteria that is rigid or flexible because their peptidoglycan layer is less, comparatively less in gram negative bacteria. Result of enzyme digestion. So if we digest the gram positive bacteria, then we will see peptidoglycan less gram positive bacteria. and that form will be called the protoplast. And if we treat uh, enzyme digestion in the gram negative bacteria, then we will see the cell wall less or the peptidoglycan layer less gram negative bacteria, which is known as spheroplast. So here we will see the outer membrane and inner membrane, outer membrane and inner membrane. Okay, but in protoplast we will see only the inner membrane or cytoplasmic membrane. Okay, so this is the main difference, that is the protoplast for gram positive bacteria, spheroplast for gram negative bacteria. Next thing is sensitivity to dyes and antibiotics. And gram positive bacteria is most sensitive because their peptidoglycan layer is high or very thick, so they are more sensitive to antibiotics which are effective to peptidoglycan layer. And the gram negative bacteria is moderately sensitive because their peptidoglycan layer is less or thin, that's why. So the main example of gram positive bacteria and gram negative bacteria is gram positive bacteria means Staphylococcus aureus, this is a one example, and gram negative bacteria Escherichia coli is an example. Okay, so move on to the next slide, that is antibiotics on bacterial cell wall, how they affect the bacterial cell wall. The first antibiotic which are going to discuss, that is the penicillin. So penicillin have a beta lactam ring which bind with the transpeptidase enzyme and inhibit its cross-linking by the glycine penta bridge activity and prevent the new cell wall formation. Okay, so how penicillin work, now we will see. So this is a peptidoglycan layer, this is the tetrapeptide, and this is the another peptidoglycan layer, this is another tetrapeptide, and transpeptidase will come to cross-link between the fourth and the third amino acid with the pentaglycine bridge. But now penicillin will come and bind with the transpeptidase, and ultimately no cross linking will be done here. So what we will see, we will see all this peptidoglycan layer will be disassembled from each other, okay, like this. So ultimately what we can see, the peptidoglycan layer will be destabilized and the cell wall will be destabilized like this, and cell will be ultimately dead. Okay, so in this way penicillin can work on gram positive bacteria, okay, or on the cell wall. Okay, so now we will see the next enzyme, that is the next antibiotic, that is the lysozyme. So lysozyme is a glycoside hydrolase, that how they break the peptidoglycan layer. So they catalyze the hydrolysis of 1,4 beta linkage between N-acetyl muramic acid and N-acetyl glucosamine. So here we will see, this is the NAM structure, this is the NAC structure, and with the first carbon and with the fourth carbon, this one, and this is the beta OH. So their linkage is beta 1 4 glycosidic linkage. And what lysozyme is done, lysozyme break this linkage, okay, and ultimately NAG and NAM will be separated from each other, and ultimately the peptidoglycan layer will be break, as the peptidoglycan layer is composed of NAG and NAM. Okay, so in this way the lysozyme can break the bond or break the peptidoglycan layer. And one thing we have to remember, that penicillin break the cell wall when the cell wall is in synthesis period, because after synthesis, of the peptidoglycan layer, or after the synthesis of the peptidoglycan cross linkage by the transpeptidase, then penicillin have no work, because penicillin have to bind with the transpeptidase and provide the inhibit the cross-linking. But if the cross-linking have already done, then we have add the penicillin, then there is no inhibition of the cell wall. So penicillin work when the cell is in synthesis period, but lysozyme will work after the cell wall is synthesized. When the cell wall is synthesized, then lysozyme came and break the bond or linkage between the NAG and NAM, that is break the beta 1,4 linkage. Okay, so in this way lysozyme will work. Now we will see the archaeal cell wall structure. So archaeal, what is the difference between bacterial cell wall structure and archaeal cell wall structure? So we have seen that bacterial cell wall structure is made up of peptidoglycan layer, but the archaeal cell wall structure is made up of pseudomurein. Okay, and peptidoglycan layer, which is made up of NAG means N-acetyl glucosamine and NAM N-acetyl muramic acid, but here in archaeal cell wall, archaeal cell wall, the pseudomurein is made up of N-acetyl glucosamine, that is also present in peptidoglycan, but one thing is missing, that is N-acetyl muramic acid. In place of uh, N-acetyl muramic acid, they have N-acetyl talosamurinic acid. Okay, and another difference is in peptidoglycan layer, the NAG and NAM are bound to each other by beta 1,4 linkage, but here the pseudomurein, the N-acetyl glucosamine and N-acetyl talosamurinic acid are bound with each other by beta 1,3 glycosidic bond. Okay, so this is all about the archaeal and bacterial cell wall structure. Next we will see the cell wall less bacteria. So cell wall less bacteria, two types, mainly two types we can see. That means the one thing is cell wall deficient bacteria and another one is cell wall less bacteria. Cell wall deficient bacteria means they already in their generation of the cell, they have cell wall in their structure, but after digestion with the any enzyme or any antibiotic, they lack the cell wall, that is the cell wall deficient bacteria. And another one is the bacteria which do not have in their life, in their whole life, they do not have any cell wall, they are cell wall less bacteria. Okay, so two types we will see. So first one is the cell wall deficient bacteria, which are known as L form bacteria. So suppose this is a gram positive bacteria which have a cell wall in the outer side of the membrane, and one is the cell membrane. So the cell membrane and outer side is the cell wall. So if we treat a chemical to them, the peptidoglycan layer will be lost, and we will see the peptidoglycan layer less bacteria or cell wall less bacteria which have only cell membrane. So this type of gram positive bacteria which do not have peptidoglycan layer or lack the peptidoglycan layer, they are known as protoplast. But in gram negative bacteria, where a outer membrane is addition with their peptidoglycan layer and cell membrane, if we treat them with the chemical, then the peptidoglycan layer will be lost and they only have the outer membrane and cell membrane. So this type of bacteria, this type of gram negative bacteria which do not have the peptidoglycan layer or cell wall, they are known as spheroplast. Okay, so they are cell wall deficient bacteria, and they are called L form of bacteria because the L form of bacteria first discovered by Emmy Killenberger Nobel, who discovered or isolated them and named the L form because after the Lister Institute in London where she was working. So for the L, it means Lister. He denoted it as Lister or London. Okay. So this is the L form of bacteria, and the last thing is cell wall less form. Okay, so few bacteria are able to live or exist without a cell wall. So they are the mycoplasmas. Mycoplasma is an example of a group of bacteria that lack cell wall. So this mycoplasma have sterol-like molecule incorporated in their membranes, and they are essentially inhibitant of osmotically protective involvement. So osmotically protect, to be protected by the osmotic shock, they have a sterile-like structure in their membrane. So that's why they survive in the environment. Okay, so these are all about in this lecture, that is the lecture 3 of basic microbiology. So thank you for watching this video.